Good morning, class. This is Mr. Gee. Sorry I cannot be there today. As you can see, there is a substitute teacher. I expect everybody to be on your best behavior as always. You know what my rules are and expectations. Yesterday, to keep us on track with everything, I handed out a piece of paper yesterday to everybody in first hour. Second hour, you got all of the information today. So I want second hour to continue working on all the information. You should be familiar with what I am asking, but in case you are not, then follow along with the video, and I'll go through each of the worksheets that you're going to have. First hour. This is the worksheet here that you guys got yesterday. Okay, so it had, let me back it up. There we go. So this worksheet that you got yesterday had all the circles on it. Your job is to go through on page 141 out of your textbook and draw the stages of mitosis, starting with interphase, which is going to be up in the very top right hand side of the corner. There you go. Starting with interphase, working your way on around into prophase, metaphase, anaphase, telophase, and then lastly, the last step is cytokinesis. So just a quick reminder for you guys, that is on page 141. Make sure that you draw the arrows and make sure that you are also writing the descriptions of each step that are found in each of these different phases. There's a difference in just copying it directly out of your textbook versus what I want you to do is I want you to read it out of the textbook, and then I want you to understand what it's talking about, and then I want you to put it in your own words, describing each step that is on the paper. So once you have this complete, again, I do want it colorful. I want you to show all the different DNA. The colored pencils should be on my front table already. If they are not, many of you guys know where they're at. They're in the waves, or the cabinet with the waves poster on it. So do make it colorful. That's one of the biggest things with it, of how you're going to get the points in this. The second thing, first hour, that you're going to get a packet that is stapled together. In this packet, you're going to have, this is going to be the very first page. Hopefully this first page is familiar with everybody. This is what was on the second part of your test. So more importantly, what I want you to do today is you need to go through and you need to fill out these, all the different parts to the cell cycle. So starting with the gap 1 phase, moving over into the synthesis phase, and then the last or third phase moving into the G2 or the gap 2 phase. The other thing I do want you to keep in mind is that collectively from this point right here all the way around to this point right here, collectively it is all known as interphase. So all three of these phases make up the interphase. This whole middle section, for those of you that are still watching, this whole middle section right here that I am highlighting right now, this is where mitosis is going to take place. So we have mitosis down here. This is what's taking place there. This is where you're going to put in your different steps to the mitosis process. So this is where we can depict that most of the cell is going to spend about 75% of its life cycle in interphase. Again, going through here, this is where it's growing. The S phase is the synthesis phase where the DNA is being copied. G2 is where it continues to grow, and it goes through the checkpoints along the way to make sure that it can undergo and it's ready to proceed to the mitosis stages. So from there, when we get to mitosis, we go through the different PMAT stages. It's just my way of remembering it, but PMAT, we have M, A, T, and the very last stage is C. So PMAT stands for the prophase. Then M stands for metaphase, A is anaphase, T is telophase, and then the C is cytokinesis. So you do need to remember that the main part associated with cytokinesis is cyto. Okay, so that is the part that splits the cytoplasm directly. Remember that mitosis is the splitting of the nucleus itself, and cytoplasm splits just the cytokinesis splits just the cytoplasm of the cell. So you do need to go through and label all those different parts, be familiar with it. That's going to be on your retest that we take on Friday. So again, if you haven't started studying yet, you probably need to start doing that, looking over everything now. I know some of you guys are going to be gone tomorrow for band and various other things. My band people that are going to be gone, you're still going to be expected to take the test when you come back in on Friday. All right, moving on to the second page of your stapled packet. The second page of your stapled packet is going to look like this here. This is where, if you guys get a chance, if you look over by the wall that's under, look over by the wall on the table, I have 3 microscopes that are set out. Everything's already focused in place. You don't need to touch the coarse focus knob. The only thing I want you guys to do is to go over there, and you need to at least look in each one. The microscope on the left has an onion root tip in it. That's going to be very similar to this picture that you're seeing right here. I do want everybody to get a chance to look at it. It's really neat. This is going to be probably one of the only times you're actually going to get to see up close these cells and see their DNA in the different stages of the mitosis. The microscope that is on the right shows the different stages of mitosis. It's not near as it's not as close up as this picture is, but you can still see the different parts of the DNA. You can see it working around, but that's in an animal cell. So the onion root tip is the microscope on the left. That's your plant cell. The animal cell is the one that's on the right. Mr. Mauser has set these up, so thank him for whenever you guys see him. What I want you to do on this page is you're going to probably have to use a pen, not a pencil, because it's going to blend in with everything on the background. But you're going to go through each one of these cells. Yes, that's correct. Every cell that's on this paper. And you're going to identify the different stages of mitosis. So the very first stage here, if I look at the very first cell that comes to mind right here, if I look at that, everything's lined up in the middle. So I would say to me that looks like it is in metaphase, where they are all lined up. If the DNA is not quite visible, then I would say that the cell is still in the growing process. So you can go through here very easily and you can depict, okay, well this one here is going to be an interphase. This one's 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 an interphase. Okay? Interphase, 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 interphase. Once it becomes visible and your DNA becomes darker and everything, and you can start to see your chromosomes, then that's where we can say, okay, well, this is probably starting prophase. This one's starting prophase. This one's starting prophase. This one's probably prophase, prophase. And then from there you can start getting into which one is going to be your metaphases, where they're lined up in the middle. So here's your metaphase here. Anaphase is where they're starting to split and be pulled to the different sides of the cell. So I would say that this is going to be an anaphase. Here's an anaphase, anaphase. Telophase is when it reaches the 2 different sides of the cell. So I would say that this one here is a telophase because this is where it's reaching and starting to kind of break apart. Our nuclear envelope is going to start to form back around everything. The spindle fiber is going to break back down. And then right after telophase is where cytokinesis is going to take place. This is where we're going to have 2 new cells. So here's a good example of telophase here, and then it will split. If I look and I have 2 cells like I do right here, you can barely see the wall here in between, but these both collectively just went through cytokinesis. So now both of those cells will go back to the interphases, and they will start growing to duplicate themselves again. So I would say it's the same way with these 2 cells here. They probably just went through cytokinesis. These 2 cells here probably just went through cytokinesis. The larger cells that are up here, I'll do it in blue here, these 2 larger cells here have grown through all 3 different stages of interphase, and they are now getting ready to start the prophase. So they're at the very end of interphase. So I would say that these are in the prophase mode because they've grown to a specific size, and just like this other one that's up here in the top right corner, the DNA is becoming visible on that. So that's going to be another prophase. So that's what I want you to do. I've done about half the cells. I want you to go through and do the other half on this. Again, make sure you look at the slides that are over on the microscope as well. The other page that you have on this, the third page, says mitosis sequencing on it. This one's very simple. Is all you're doing, you're going through each of these and you're telling me what's happening in each step. So in the very first step, I'll help you guys out with this one. The DNA or the chromosomes are not visible. So here you can see that we have our nucleus. This one here is going to be in interphase. Okay, it's growing. So your options is you're going to have interphase, you're going to have metaphase, metaphase, prophase, anaphase, telophase, and cytokinesis. So you need to go through and identify each of those. I'll tell you here on the fourth row down, okay, I'm going to be looking at this box right here. I now have two new cells. This is at the very end of mitosis. This is what mitosis creates. It creates two identical daughter cells. So these two identical daughter cells are identical to the very first cell that we started out with. So after cytokinesis has taken place, such as down here in the bottom right corner, cytokinesis, now both these two new daughter cells, okay, so these are daughter cells, they're going to go forward to go back through the interphase stages. So I would say that those are back in interphase. So that's what you're doing on this worksheet. I want everybody working on that, and it needs to be done probably by tomorrow. Tomorrow I do have a yarn project that we're going to do where you guys are creating the different sequences on a piece of paper by cutting yarn and showing shapes and differences and how the DNA is being duplicated and pulled to the different sides of the cell. The very last thing I have for you, yes, everybody's favorite thing, is another color page. Okay, I do like the color pages. They explain a lot. The only thing that I want you to do on this color page, though, specifically, is just the mitosis part. Okay, do not worry about meiosis. That does not mean cross meiosis out, because we're going to get to that part. So the only thing I want you to do today is do what's in this blue box here. Okay, just the mitosis part. You are starting with the parent cell and working your way down to the two daughter cells. So you need to color everything. It tells you what to color, the different parts of mitosis, specifically dealing with the chromosomes. It gives you the numbers there and everything. So pretty self-explanatory. These are all the worksheets that you should get. This last page, that's the color sheet, is going to be a separate page that's not in the stapled handout. So everything is sitting on the table. You need to make sure that you get each one of these pages by the end of class to make sure that you have them. If you're going to be gone tomorrow, everybody needs to work on these because these are all due on Friday. With that said, I plan to be back tomorrow. If you have any questions, Mr. Mauser is in their second hour. I will be there before school on Thursday to answer questions. We are going to work on this some more on Thursday, along with the yarn project. But all this is due on Friday. Along with that, your comp books are also due on Friday. So, don't forget, we have our projects. Okay, that's all this stuff that we're doing. The comp book. And then we're going to have the yarn project that are due on Friday. The yarn project due Friday. I will also send out a remind to everybody to let you know what you would need to keep up with the next couple days. So, your test on Friday, I do expect you guys to score a lot higher. I'm shooting for a class average of right around I would predict an 80 to an 85%. So, all right, thank you guys. Have a good day.